Chapter 3 Energy, Chemical Reactions, and Cellular Respiration. Part 3 Cellular Respiration. First of it is to make ATP, that chemical energy that our cells love to use, from food, specifically glucose, but food. It is an exergonic multi-step metabolic pathway, so many steps, many enzymes involved. The whole purpose being to release energy that can be used to make ATP. There are organic molecules that get oxidized and disassembled. It's a long, complicated process. And then potential energy in chemical bonds gets released. So again, the whole point being to make ATP, because ATP is the energy currency of the cell. This process requires oxygen. This is why we breathe in. And it produces carbon dioxide, which is why we breathe out. So it starts, well, the whole process is referred to as glucose oxidation. We are burning glucose, basically. Long step-by-step process, turning glucose into carbon dioxide and water. So the overall chemical reaction, glucose plus 6 oxygens will yield 6 carbon dioxides plus 6 waters plus energy. That'll become ATP. So, turns out that some of the energy is made by breaking bonds and capturing phosphates and attaching them to ADP. This is substrate level phosphorylation. So, we have this thing with a phosphate group and an ADP molecule, and as it changes into pyruvate, ATP is formed. There's not a lot of this that occurs in cellular respiration, but there is some. There's some. What primarily happens is energy is transferred indirectly. So the energy is released to a coenzyme, either NAD plus or FAD, and these coenzymes will then carry the energy to get used elsewhere to form ATP. And it's called oxidative phosphorylation. Oxidative because oxygen is involved, phosphorylation because we're adding phosphate to the ADP to get ATP. And this is where most of the energy comes from, this indirect transfer. Stages of glucose oxidation, or you could say stages of aerobic respiration or cellular respiration. We have glycolysis, intermediate stage, citric acid cycle, and electron transport system. Glycolysis is anaerobic, doesn't need oxygen, occurs in the cytosol, outside of the mitochondria, and it does not need oxygen. The intermediate stage, citric acid cycle, and electron transport chain all occur in the mitochondria, and they're going to need oxygen, and they're the aerobic part. So, boop-a-doo-doo. Anaerobic cellular respiration, glycolysis occurs in the cytosol. Aerobic cellular respiration, those 3 stages occurs in the mitochondria. Glycolysis, first part. Glycolysis is anaerobic. Glucose gets broken into 2 pyruvate molecules. We start with glucose, we end with 2 pyruvate molecules. And pyruvate's fate depends on oxygen availability. During this glycolysis, breaking down glucose, we get a net production of 2 ATP and 2 NADH molecules. It is a 10-enzyme step. So we start off with glucose, lots of things happen, lots more things happen, and then we get pyruvate, 2 pyruvates, and a net gain of 2 ATP and a net gain of 2 NADH. So, summary, we start with the glucose, we end with 2 pyruvates, which are 3 carbon molecules, and 2 ATP and 2 NADH, or NAD plus gained hydrogen. What happens to pyruvate? Well, if there is sufficient oxygen, it will go to the mitochondria. If there is not enough oxygen, pyruvate gets turned into lactic acid. This is a type of fermentation. So glucose becomes 2 lactic acid molecules and 2 ATP, and that's it. This is inefficient. It sucks. We don't want to ferment, but sometimes it happens, especially say in our muscle cells if you're working out really hard. Intermediate steps. So the pyruvates, two of them, enters the mitochondria, technically mitochondrion, and the intermediate step is what's necessary to get it to the citric acid cycle. So what happens? Pyruvate combines with coenzyme A to form acetyl coenzyme A. So pyruvate plus coenzyme A leads to acetyl coenzyme A, which is a 2-carbon compound. Also, we get an NADH formed, and the carbon dioxide produced, decarboxylation, losing that carbon to form carbon dioxide. Then we move on to the citric acid cycle. That acetyl coenzyme A is going to enter the cycle. It's 9 enzymes involved, occurs in the mitochondrial matrix, and the acetyl coenzyme A ends up producing carbon dioxide as well as ATP, 3 NADH, and 1 FADH2. Of course, we have 2 acetyl coenzyme A's going in because we had 2 pyruvates, so it's really doubled. Here is the whole process. It's complicated and crazy. We're not going to worry about the whole process, but you do need to know that the beginning of the process is when oxaloacetate combines with acetyl coenzyme A to form citrate, hence citric acid cycle. Go through all these processes, and oh look, we remake. The final product is the same thing as the initial substrate, oxaloacetate. So, summary of citric acid cycle occurs in the mitochondria, requires oxygen. You need to start with acetyl coenzyme A and oxaloacetate. We produce 2 carbon dioxides and 1 ATP, 3 NADH, 1 FADH2 per cycle. Since there's 2 turns, 2 acetyl coenzyme A's are going through the citric acid cycle, we actually get a net production of 2 ATP, 6 NADH, and 2 FADH2s per glucose molecule. So that is the chemical breakdown of glucose into carbon dioxide. So our 6-carbon glucose becomes 6 carbon dioxides that we breathe out, where we're gaining ATP and NHD and FADH2 during this whole complicated process. From here, we move on to the electron transport system. Electron transport system is a series of enzymes found within the inner membrane of the mitochondrion. And in the process, energy is going to be released to form a hydrogen ion gradient. And that gradient will be used to make the ATP. Again, hydrogen ion gradient, lots of hydrogen ions on one side of the membrane, very few on the other. That's a lot of potential energy that then gets used to make ATP. And again, all the important enzymes are found in the inner membrane of the mitochondria. So, important structures for electron transport system is all those proteins that catch electrons from the NADH and the FADH2. So NADH and FADH2 release their electrons to the electron carrier proteins. Proteins then act as a hydrogen ion pump, so that you end up with lots of hydrogen ions on the outside of the inner membrane, very few on the inside of the inner membrane. This produces the hydrogen ion gradient. That is a lovely pool of potential energy. And then ATP synthetase takes advantage of this hydrogen ion gradient to produce the ATP, to add phosphate groups to ADP to produce ATP. So the ATP synthetase is the enzyme that produces most of the ATP. So, process. Electrons first transferred from NADH and FADH2 to oxygen. That's right. They release hydrogen and it becomes oxidized. Electrons are passed to the oxygen O2, leading to the production of water. So we have oxygen combining with electrons and hydrogen ions to produce water. This is again why we breathe in, is to make energy, to use the energy found in NADH and FADH2. A protein gradient is established. Again, those carrier proteins are going to form that proton gradient proton gradient, hydrogen ion gradient, same thing. And electron energy will be used as kinetic energy to pump the hydrogen ions or protons out, forming the gradient. And then the ATP synthetase will take advantage of that gradient, allow hydrogen ions to enter again inside the matrix of the mitochondria. So as the hydrogen ions, the protons move down the gradient, they're turning the wheel of the ATP synthetase, as it were, in order to bind phosphate to ADP to get ATP. And this is oxidative phosphorylation. Oxygen accepts the electrons. ATP is formed by ADP being phosphorylated, oxidative phosphorylation. And this is different from the substrate level phosphorylation we saw earlier in this whole process. That all the times ATP was produced in glycolysis and citric acid cycle, those were all examples of substrate level phosphorylation. Oh, right, it turns out cyanide binds to one of the important proteins involved in the carrying of the electron and forming the hydrogen ion gradient. So this inhibits the transportation of the energy, inhibits the production of ATP. So cyanide is basically making our cells starve for ATP energy because the cyanide is breaking this pathway, preventing it from occurring. Electrons cannot reach the oxygen. And so to try to deal with that, you have to take medications that will bind to the cyanide to try to pull it away from its normal target. All right, so this is the breakdown of cellular respiration. ATP being formed from the breakdown of glucose. Glycolysis gives us 2 ATP and 2 NADH, which can become 6 ATP. Intermediate stage gives us 2 NADH that can become 6 ATP. Citric acid cycle gives us 2 ATP and 6 NADH that can become 18 ATP and 2 FDH2s, which can become 4 ATP. Grand total substrate level phosphorylation gives us 4 ATP. Oxidative phosphorylation gives us 34 ATP. And then, unfortunately, we have to use a little bit of ATP during glycolysis. Got to start the process. So basically, we end up with 36 ATP. So the final formula you're going to want to remember is 1 glucose molecule plus 6 oxygens. Molecular oxygens lead to 6 carbon dioxides plus 6 water plus 36 ATP. Wow, aerobic cellular respiration is so much better than fermentation. So much better. Okay, it turns out that other food sources can be used besides glucose. You can use other monosaccharides. Often they get turned into glucose. You can use fatty acids. Muscle tissues like using fatty acids from our lipids. And basically 2 carbons are removed at a time to form acetyl coenzyme A that then enter the citric acid cycle. And we can even use proteins. You see, the amino acids can enter the pathway, although they enter in different places within the pathway. So again, we can get energy from our fats, fatty acids. We can get energy from the proteins, the amino acids. These are all energy sources besides the carbohydrates. All right, that is it for this part of the lecture. I hope you enjoyed chapter 3.